Hello, everybody, and welcome to Nano at Tech, the webinar series sponsored by the Institute for Electronics and Nanotechnology at Georgia Tech, the Southeastern Nanotechnology Infrastructure Corridor, which is a member of the National Nanotechnology Coordinated Infrastructure. So it's a pleasure to have with us Professor Amy Herr from University of California, Berkeley. Amy got her bachelor's degree in engineering and applied sciences at Caltech, then moved up California to Stanford, where she earned her master's and PhD in mechanical engineering, and then spent some time at Sandia National Labs in the Biosystems research group before going to Berkeley, where she is currently professor of bioengineering and Chan Zuckerberg Biohub investigator. She's won a number of research awards, including NSF Career, NIH New Innovator, Sloan Fellow, and DARPA Young Faculty Award, as well as being an elected fellow at the American Institute of Medical and Biological Engineering, and a member of the National Academy of Inventors. Turn it over to Amy and turn off my mic so dog number 2 doesn't disturb us too much more.
Thank you. And can you hear me?
Yes, I can hear you.
Okay, I was waiting for a dog to bark. Excellent. Fantastic. Well, thank you all for joining today, and thank you for the invitation to come. It's an honor to virtually be here and share with this community, which I'm just really intrigued by the program that you've set up and the community that you've set up. And it's just lovely to be able to interact with you. Of course, I wish I could meet with you in person, but we look forward to getting to that day sometime in possibly the near future. So thanks for your time today. Again, my name is Amy Herr. I am a professor at UC Berkeley, also affiliated with Lawrence Berkeley National Labs, and have had the pleasure to be involved with the Chan Zuckerberg Biohub for the last couple of years. So today, with this particular audience, I wanted to really focus in on how different innovations and approaches to microsystems design can really help advance biomedicine and the biosciences. So for those of you in the audience, if you have a background in nano and micro, which I assumed would be a large fraction of this audience, what I'm hoping to do is to be able to show you how the choices we make can help open up new avenues for measurement, hopefully meaningful avenues for measurement for those that we collaborate in biomedicine and the biosciences. So again, thank you so much for the invitation today. I have just a couple of disclosures not related to this work that are listed at the bottom of the slide. My group is really focused on measurement gaps. So we are a measurement science group, as I mentioned, in the Department of Bioengineering. We have focused on gaps in measurement science, in biological sciences, that we feel are important, but also neglected. One of these major areas is the direct measurement of unmodified endogenous protein targets from single cells. Of course, probably most of you are quite familiar with the impact that measurement science and new tools has had on single cell genomics and transcriptomics. Those two areas are just exploding in terms of the number of new approaches and the power that the ability to measure directly DNA, RNA through sequencing in single cells is bringing to the biological sciences and also biomedicine. Of course, if we use the central dogma as a guide, we know that DNA and RNA are important, but we also know that the end products of that central dogma, protein and protein expression, literally are the molecular machines of the cell. So we have information encoded in the DNA and the RNA, sort of like blueprints of life, if you will. And when we think about proteins, we think about those as not the blueprints, but those molecular machines that are doing the work of responding to the local environment through cell signaling. So if you're not an expert in protein science, that's all right. I just want to give you a little recap here on a specific huge area that's been literally a blind spot to the central dogma and to single cell biology. And that is this area that has recently been called proteoform science. So essentially, when we think about protein targets, We oftentimes think about a protein target as manifesting as a single molecular type, but that is actually not the case. In fact, that is very, very far from reality. When we think about proteins, we think about those proteins coming from the human gene, being expressed as a particular species, a particular molecule. But in fact, what often happens is there are different variations of that single protein molecular type that can be expressed. And that's because of this process that results in isoform formation, so different forms of that same target that have slightly different properties, everything from conformation all the way to differences in their amino acid sequences, slight differences. We also have post-translational modifications. So here on this slide, listed as site-specific features. So these can be things like decoration with phosphorylation, decoration with the glycosylation, just all of these different types of molecular modifications that happen to a protein target. And of course, there are literally thousands to millions of these targets expressed in each cell. So each target's type can have multiple molecules of that single type. And within that molecule, there is this huge distribution of how that protein target, each individual molecule is being decorated with different post-translational modifications or modified in different ways. And this leads to proteoforms. I'll give you a couple of examples of proteoforms, but suffice to say, when you think of a protein target expressed in a cell, just remember that target is actually an entire distribution of different forms of that same molecule. These forms are pretty important to cellular function. And again, I'll give you a couple of examples there. Just to give you a sense of the scale of this blind spot, I'll call it in biology, when we think of protein encoding genes, we think there are about 20,000 of them. And that's really coming from the great work of the Human Genome Project. When we think about these different forms that can be expressed, the protein forms that can be expressed, there are about 70,000 of these different forms that can be expressed. And that's something we call a splice variant when we're thinking about alternative splicing. But when we start to think about the ways that each of those molecules can be individually modified, we start to think about this concept of proteoforms, and we start to see that that number of expected proteoforms is absolutely huge. So I add two references here that make the valiant effort to try to estimate the number of these different forms of proteins that exist, and that number quickly exceeds 10 to the 9th. And so this is a huge measurement science problem. A lot of the advances in nucleic acids analysis, single cell resolution, so sequencing, really come from the fact that we're talking about molecules that are built of nucleic acids. These are really well-controlled, well-behaved, maybe is the better word, groups of molecules that come together. When we think about proteins, we think about amino acids, so many more of those amino acids, so much more physicochemical diversity among those molecules. And then on top of it, we have this added modifications that happen just through the processes of life and disease within an individual cell. So if we think about that number, 10 to the 9th of these proteoform targets probably exist. We ask questions like which ones are important, at what levels are they expressed, and then we start to think about things like spatial distribution within an individual cell or a tissue, subcellular localization. And then, of course, we know that inside of a cell is not homogeneous. It is not uniform concentration. So we know that concentration gradients also exist. So this is a really, really rich area for those of you who are interested in looking at bringing quantitative capacity to biology and biomedicine. And I'll give you a couple of examples today of how my research team at Berkeley has started to tackle this using microsystems design. So just a little bit additional primer background in terms of what exists. As an engineer, we oftentimes like to do this benchmarking analysis to understand what is out there, what can these tools currently do, and where are there important gaps, what I'm calling unmapped areas in our atlas of biology and disease. Some of you may be familiar with the human cell atlas projects that are going on right now, trying to understand the different cell types and states that are present in biological systems. So I mentioned sequencing. I do obviously want to say that these are really powerful tools for measuring DNA and RNA, again, even at the single cell level. But as I touched on the previous slide, sequencing of nucleic acids does not reflect the dynamic protein expression, modifications, and we haven't even talked about protein-protein complexing that happens. In addition to that, we have tools like mass spectrometry, exceptionally powerful for direct measurement of proteins. I won't get into it too much today. We are making headway with pushing down detection limits to get to single cell and subcellular detection capabilities. We do still have limitations on throughput. And then, of course, the mass spec tools, if you're an aficionado, that are making the most headway towards single cell measurements are these bottom-up approaches that rely on protein digest to peptides to be able to make their exquisite measurements of proteins. Unfortunately, that basically scuttles all of the information about the stoichiometry, or what's the distribution of each molecular target that's present. And that's exactly what we're looking at with proteoforms, so that's an issue. And then the last one, the de facto standard, the powerhouse for direct measurement of proteins in individual cells, is the humble immunoassay. You may be familiar with benchtop approaches like ELISA, immunofluorescence. If you've ever seen a pathology slide, immunohistochemistry, looking at a tissue slice is a great example. Newer approaches like mass cytometry and, of course, flow cytometry are all immunoassays. Here, we are limited by the availability of immunoreagents. So the antibody probes that we use to detect the presence of those a priori identified targets. This is a targeted proteomics method, which is really important when addressing biological hypotheses. But if that immunoreagent doesn't exist, or if it's not selective enough to distinguish between different proteoforms, again, modifications to a single protein target that let that form be expressed in a whole distribution of molecular types. When we look at single cell measurements, we can also get confounding background signal that really limits our ability to target multiplex. I just want to call your attention to this little pie chart over on the left. I've put a black arrow on that to show you this tiny slice of that pie chart that really shows how many probes exist for proteoforms. And the number that's estimated right now is well under 1% of proteoforms can be detected with an existing antibody reagent. Most immunoreagents are developed for genome encoded proteins. So that just gives you a sense of how big that blind spot is. So the approach that we've used is recognized these powerful tools that exist, and we've asked ourselves, how can we augment and expand measurement science by bringing in new approaches? So for us, that means designing for single cell and subcellular resolution. It means designing tools that have a selectivity that can get us into this molecular heterogeneity, looking at these different proteoforms. And also, in addition to that, it says, hey, a lot of these immunoreagents are out there. How can we make those more powerful? Well, also designed to be functional with any sort of new detection reagent like aptamers and others that come online. So these are really kind of the specifications we're using to meet this quest of mapping these unmapped areas in our Atlas of Biology and Disease. So how does this interface with microsystems and nanosystems? Thanks so much for those of you who are MEMS engineers, as I have been as well, kind of thinking about what is this problem, what is this area, as a designer saying, what are these unmet needs, and now coming in and saying, okay, we understand the problem, the scope of the problem, the need. What is one approach to being able to provide solutions? And for us, that has involved being able to isolate single cells for isolation and manipulation, sample preparation, which I would argue is one of the unsung heroes of measurement science, is oftentimes needing exquisitely precise, timed and chemistry-wise manipulation, and being able to do that at the single cell level is pretty powerful. We have chosen to marry these single cell manipulation methods, in our case, microwell arrays, And you may be familiar with droplet-based methods and other methods of compartmentalization. We've chosen to marry those with separation science. And for us, we just love electrophoretic modalities, methods that can use an applied electric field to be able to distinguish species based on differences in their characteristics, be different sizes of the proteoforms or isoelectric points of the proteoforms or charge state, complexing, all of these different capabilities. We add that separation step of the molecular constituents of a cell to be able to pull apart this physicochemical properties of interest. And we bring that to bear with this last step, which is the humble immunoassay. So for us, instead of measuring just one property, the absence or presence of binding to an immunoassay probe, an antibody probe, we also have another piece of important information, and that's this physicochemical property of each molecule that we choose to measure, which means that we have higher specificity, that selectivity I mentioned earlier, than the immunoassay alone. And we do this by using microsystems design. Our overarching goal is to control the chemistry of what's happening in our devices in space and time. And we use that using mostly passive methods, so you won't see any pneumatic actuation, pumps or valves on our devices. We primarily rely on bulk approaches and diffusion, as well as electrophoretic transport. And we do this using open microfluidic frameworks. So for those of you who are MEMS designers thinking about biomems, you've probably seen some of these approaches before, but I hope to highlight the power and the trade-offs of open microsystems design to give you something to think about, either for your own work or for our field more broadly. So let's get specific. What does this look like in terms of the avenues that my group has been pursuing? We have chosen again to use these microwells. So what you're looking at, and I'm not sure if you can see my cursor. I should have asked that earlier, but if you can, what I'm circling are two GFP, so green fluorescent protein expressing mammalian cells settled into a microwell in the center of the screen. So that red material that you see, that is a rhodamine infused polyacrylamide gel. So this is a hydrogel material that is quite often used by conventional tools in biology that we've adopted as one of the approaches, the materials that we like to use for the biomems devices that we develop. We've done a couple twists on it that I'll tell you about, though, to make it more functional for these integrated devices. But as I mentioned, we settle individual cells, or in this case, a pair of cells, into that microwell. It's a cup that's been molded into this hydrogel, surrounding hydrogel. So if you look down on the system, so on the right-hand side of the slide, you see single cell in a microwell. This is just a cartoon drawing, obviously. You see a circle, and that's the microwell, and you've got that little dashed darker gray item in the middle, and that's a cell that's ostensibly settled into that microwell. After we isolate those individual cells, we then lyse. So that's the sample preps part I was talking about. We break open that individual cell, spewing the contents of that cell, that lysate, into the microwell geometry. The beautiful thing for us about the types of systems, these open systems, and I'll show you some bright field images in a couple of minutes, is that we can very, very quickly apply an external electric field. So you see that here with the electrode symbols, causing the lysate that was housed inside that microwell to now electromigrate through the wall of the microwell and into the surrounding polyacrylamide gel. So that polyacrylamide gel is a structural material. It forms the microwell. It forms the substrate in which the microwell is cast. But it also, once we apply this electric field, acts as a molecular sieving matrix for electrophoresis. So we like to use these materials to do double, triple, or quadruple duty. And this is one example of polyacrylamide hydrogel as a structural material for that cell isolation, and then also as a sieving, molecular sieving matrix for that first step, which I mentioned earlier, the separation, to get out information about the physicochemical properties of those proteinaceous compounds that are in the lysate of that cell. And here you just see two peaks. Those are two different protein species that are separating from each other. Probably the first one to the right is the fastest, meaning it's probably the smallest species, and it's resolving from that larger spot, which indicates the maybe a smaller species. So we've completed the separation now. What we need to do is add that immunoassay. And this is a little bit of a challenge because in this case, at the separation stage, we're working with a hydrogel. We have pores in that hydrogel that are filled with buffer water. It's an aqueous system. So we've got the molecules of interest, they're resolved in space, but they're still diffusing. It's a homogeneous system. Nothing is tacked down. Everything is in solution phase, albeit within the hydrogel matrix. When we perform an immunoassay, we need our species, our target of interest, to be immobilized if we're going to perform what's called a heterogeneous immunoassay, which is pretty helpful for us because you'll look at the length scale on the far right of this red confocal micrograph material that I showed you in the center of the screen. The size of these microwells is about 100 microns in diameter. So all of you are familiar, that's about the diameter of a human hair. And when you think about these protein species now being liberated from the cell, so they're no longer compartmentalized, they are diffusing. And as you probably realize, diffusion over these tiny length scales of microns is actually very efficient. For someone who's trying to complete separations, diffusion is actually a big issue because diffusion works to disperse molecular species, which is exactly the opposite of what you want as a separation scientist. You don't want band broadening and these species continuing to diffuse into each other. So that leads us to what we do to the hydrogel material, this polyacrylamide in this case, to make it, choose to use material that's of interest to biologists and they're, commonly familiar with working with it, but tweak it a bit to give it some function that's going to help the microdevice in the long run. And for us, that means including a photoactive moiety. This is benzophenone, which we've synthesized with methacrylamide to allow that photoactive small molecule to be basically cross-linked into the polyacrylamide gel when we form the microdevice, when we cast the microdevice. Why do we do that? We do it because we can use that molecular sieving matrix for electrophoresis. And then if we expose the device to UV light for a short period of time, we activate the benzophenone and that abstracts hydrogen from all of the CH bonds that are in that hydrogel, which is great because the proteins we're looking at have a lot of those CH bonds. And we basically end up anchoring all of the protein to the hydrogel. So this basically photoactive species allows us to turn what was a homogeneous system, so those molecules just freely diffusing through the pores of the hydrogel, into a heterogeneous system where we've now got those species, the targets of interest, covalently locked into the polymer. And that allows us to then complete the next step, which is that in situ or in gel immunoprobing, so that we can then get out that important second piece of information, which is the presence or absence of target as determined by binding to our immunoassay probe. We typically use antibody probes that have a fluorescent label. We've also done some work recently using antibody probes that are conjugated not with a fluorophore, but with a rare earth metal, allowing us to use SIMS or MIDI type approaches, mass spectrometry, to be able to get a readout. And I won't talk about that today. So here's the overall. I've spent a little bit of time giving you background on this framework and some of the underlying engineering rationale for the choices that we've made. Because I'm going to use this framework for the rest of the talk today, the key thing to remember is our overarching goal is listed right here under the title, and that is to report not one, but two properties of each target, protein target that we're interested in, and that gives us enhanced selectivity so that we might be able to measure those proteoforms of interest. So you could think about this as if we have two proteoforms, they are reactive to the same immunoprobe, but they are different sizes. You can quickly see how this type of approach will resolve those two proteoforms in space and then use that same immunoprobe to indicate the presence of both of those species. So a powerful approach that's been used at the macro scale for decades in separation science. We've just brought that to the micro scale in single cell biology. This is a bright field image, a little bit of an isometric view here of what a typical device looks like that's coming out of my lab right now. They're basically functionalized microscope slides. So what you see here kind of on the right edge is you're looking at the edge of a microscope slide, and then you're looking right above it at a thin layer, kind of the bluish gray of that photoactive polyacrylamide gel. And then you can see stippled into that thin layer, about 30 to 150 microns thick, you can see these hundreds of microwells. And then adjacent, abutting each microwell is that separation lane that I mentioned earlier. So no precision actuation, as I mentioned before, an open format. There's no enclosed microchannels at all. This is basically looks just like a microscope slide to kind of first glance, which is exactly what we wanted it to do because we are pretty interested in translation. And that means developing tools that are amenable to the approaches that are currently used in either the biomedical or biosciences labs of our collaborators. It means more work for us on the front end to figure out how to impart different sensing and actuation function, but on the back end out in the translation, it means that the people we work with who are interested and familiar with pipettes and imaging using scanners and not needing large pumps and valve kind of compressed air banks to be able to run that. So just a different approach to microsystems fabrication. Okay, so I'll keep moving here with some examples, again, because the new work I'm going to show you a little bit later really requires that you understand kind of what these assays look like. I think I've emphasized the importance, the unmet needs that we've designed to meet. On the top, you have just another image of one of our single-cell immunobonding chips, again, that open structure. If we were to zoom in on one of these blocks of microwells, which we do right under that image, this is a false color fluorescent micrograph. You can, if you squint, you can see the rows of microwells, and then below each row, it looks like almost a little comet of purple or reddish color. Those are the protein peaks that I mentioned. So this is the output after the separation and immunoprobing, again with a fluorophore on our immunoprobe so that we can see it when we use fluorescence detection. And that's what you're looking at. If you zoom over to the right, I've just pulled in a bunch of bright field micrographs at the top of the image that show those individual cells, and in some cases pairs, and in some cases no cells settled in the microwells. In this particular example, we used simple sedimentation to load the cells into the microwells. If you've looked at anything like droplet encapsulation of cells, you're familiar with Poisson statistics, which basically tells us that in some cases we're going to get single cells in the microwells, in other cases we'll get none. Some cases we'll get 2 or 3. We do design the wells to bias that distribution towards single cells. If you're interested, at the bottom we have a Nature protocol from a couple of years ago that really talks about how you design those microwells to push you towards the probability of 1 cell per microwell. Right below those bright field images, you see the companion readout from the single cell. In this case, it's a Western blot. We are resolving the different targets based on differences in size. And so over on the right, we've looked for beta tubulin, which is a very highly expressed cytoskeletal protein in mammalian cells. And then we've looked for this enhanced green fluorescent protein, which is engineered into our cells for development of the assay. You can see the cells that have the beta tubulin present and those subset that have that GFP being expressed. The separation length, I want to draw your attention to the dimensions. So on the left-hand side here in kind of the center of this image, you'll see that that separation length is about 500 microns long. Now, from a separation science point of view, that is exceptionally short distance to be able to complete a separation in. If we're looking at the conventional systems that biologists use, they're using tens to centimeters of distance. So a really big kind of macro scale cards they use to perform these separations. For us, we've just shortened that tremendously. We can get away with it because of the microsystems design, the speed at which we can complete the separations. We're able to basically outrun diffusion because we complete our separations so quick as compared to the time scale of diffusion, and that's quantified through something that we call the Berkeley number. And then at the bottom of the image, you can see those traces. We've gone in and done Gaussian fitting to each of the peaks, which means that we can pull out quantitation like the estimated molecular mass of each species on the far right, And then also something that we call the area under the curve, which is the amount of material that's housed in each of those peaks that gives us an estimate of the protein expression, which is important at the single cell level. So we've published a couple years ago, so I will skip over it in detail. I just wanted to assure you that we've spent time looking at the lower limits of detection, the dynamic range of detection for this type of assay here using conventional fluorescence detection. Our system is on par with the conventional system of flow cytometry, which makes a lot of sense in terms of how each of the different tools, the physics of each of the different tools, but I'll leave it at that for today. And then just wanted to highlight a couple of our applications, these unmet needs that we've dug into with regards to this system. We have some amazing collaborators right now, and I'll show you some of the work that we've done with them. Before I do that, I just wanted to call out something that I actually thought was impossible. And one of my immensely talented and stubborn graduate students really was convinced that this was possible, and he definitely proved me wrong here. The physics said it was possible, but it just seemed a little bit too precise to be able to implement with our system, but he was able to do it again and again reproducibly. So what I'm showing you is just two things. Across the top is those fluorescence traces. So as a function of distance, kind of how much fluorescent signal are we getting out? You can see those Gaussian fits. And the image itself, this is again one of those false color micrographs. The dimensions are at the bottom, and you can see that microwell kind of right by the XY axes that has a little bit of residual cellular material in it, depending on how close you're sitting to your screen or how well this is getting to you. To the right-hand side, we have that protein separation that I talked about before. In this case, we're using a mode of electrophoresis for the first stage before the immunoprobing that resolves species based on differences in their isoelectric points. So that's the point in a pH gradient at which the species has net zero charge overall. And what you can see here is we're able to resolve here 1 and 2 in blue, 2 isoforms of a particular target. But the important thing I want to draw your attention to is the green, 1, 2, and 3. We're able to detect 3 isoforms of the green fluorescent protein that was expressed in that cell. And those isoforms are estimated to have single charge unit differences by NMR. And so this shows you that your choice of that separation modality, be it separation by molecular size, or in this case, isoelectric point, is really important in terms of being able to dig in to the property of the proteoforms that differs between each of those species. So I just wanted to include that here because it's single cell, single charge unit differences in proteoforms that are measured here using a probed isoelectric focusing assay. Most of our work shifting down to the bottom of the slide, has been pretty interested in oncoproteins. You may be familiar with classification of cancer as HER2 positive or estrogen receptor positive, progesterone receptor positive, or even triple negative. Those designations are referring to overexpression of an oncoprotein on the surface of the cancer cell. In our case, we're collaborating with Mark Pegram's group at Stanford University Medical Center. He's the director of the Women's Comprehensive Cancer Clinic. His interest has been in HER2. At the top of the slide, there's a little schematic of different proteoforms, so these red and black boxes that you can see. On the left-hand side of that little schematic, we're typically thinking about a full-length HER2 protein, and so it's a transmembrane protein. This is to represent the cellular membrane, the cytoplasm, and the cell membrane, and then outside the cell. Typically HER2, it's 185 kilodaltons, so it's a pretty big protein. It is a protein that is the first target of the first FDA-approved targeted cancer therapy. So the FDA approved several years ago Herceptin or trastuzumab, which is a humanized monoclonal antibody that comes in and latches on to this HER2 protein, now oftentimes carrying a cargo of drug that allows that therapeutic to kill the cancer cells. Well, suffice to say for today, the HER2 protein does not manifest always as this transmembrane protein. So it doesn't always have this black box, that extracellular domain that the drug latches onto. Sadly, it also, this HER2 protein manifests as different proteoforms that lack that extracellular domain. So immediately, you know, this is important because these truncated isoforms don't have the extracellular region for that drug to latch onto, meaning that these types of cells that have this high expression of the truncated forms of HER2 don't respond to therapy in the same way we would expect if those HER2 proteins were expressed as full length. So this is probably the best example from today's talk for the clinical implications. If you're interested in translation to clinical science, this is an example of where the understanding the stoichiometry of the different proteoforms is important, because it's directly going to inform the mechanism that you're disrupting with an external therapeutic. So 2 micrographs here at the bottom. We've got kind of that magenta with a purplish blue. These are the single cell separations. They're now oriented top to bottom. I apologize for that. But the key thing I want to point out is the micrograph that's labeled P185. You can see just one of those magenta peaks. That is the full-length protein. It's big. It moves slowly away from the microwell, so it's close to the top of the gel. And if you shift over and look at the micrograph that's labeled P110, you can see it's got the P185 form of HER2, but you can also see it's got this smaller truncated form. It's 110 kilodalton, so we call it P110. And you're seeing that it's detected in the same color channel, magenta, because it's being detected by the same antibody as the P185. And that's the crux of all of the work that we're doing. We can use separation science to resolve in space, separate out these 2 species, the P185 versus the P110, and then use the existing immunoreagents to actually detect those species. So here we have information on the stoichiometry. And we've extended this to circulating tumor cells. We were benchmarking against immunohistochemistry, which is the gold standard, one of the gold standards for clinical classification of cancers. And we, of course, are quite interested in other oncoproteins. And this just little image here is an example of this estrogen receptor. It has also 2, actually many more, isoforms that are of interest to us as well. So hopefully that gives you a little bit of a recap of where we are to date with really enhancing this taxonomy that is used for cancer classification as an example. So an image of our device on the right, and then on the left, we just have some false color micrographs that show the multiplexing. So this is one cell that has been subjected to multiple rounds of probing after that separation step, allowing us to really dig in to the targeted protein expression within that cell. Where are we going? I wanted to maybe share some of our more speculative work with this group, making sure to leave time for questions and discussion, because I know it's awkward on Zoom, but I am hoping that this group in particular, it's not every day I get to talk to excellent top engineers in the micro and nano field. So I'm looking for hopefully some at least stilted discussion using Zoom. But we've been thinking about ways to kind of toggle the form factor of these types of devices. So at the top, you see one of our open microfluidic devices. Again, it has these little microwells stippled all over the device, but we've also created sort of a perforated perimeter for each of those microwell plus separation links. And we did that because we can now, after we complete that assay, the measurement steps that I just mentioned before, release from the planar device into solution each of those separation lanes as a rectangular particle. And so we've done that for a couple of reasons. On the right-hand side, I'm just putting out some estimates that we're using for how we can enhance the transport of immunoprobe into the gel for the last stage of that assay. So I'm not going to talk about it today, but thermodynamic partitioning does everything it can to keep those immunoprobes, those huge antibodies, out of the hydrogel. So we're working in a difficult regime for immunoassay performance. Anything we can do to help enhance delivery of those large immuno reagents into the gel, we try to do. And we have some active measures, I'll call them, just active approaches that we use to do that, as well as passive diffusion, but reducing the length scale helps. And here is just an example of one of our release particles that you can see. It's not fabulous signal to noise, so the noise is greater in this system, but you can see each of those peaks above the SNR of 3 that are basically almost barcoded on that released microparticle. And the goal here is to be able to harness the improved physics, but also to concatenate with something like flow cytometry, so that we can then perform readouts, not with a microarray scanner and fluorescence imaging system on a planar device, which is what we do now, but with something that, again, is a piece of equipment that's widely used in the biosciences, and that's the goal here. Last thing I'll say before I jump to the final topic in just a couple of minutes, this ability to kind of go from 2D to 3D is pretty important to function, we found. And so this is just a different schematic, but it shows along the bottom exactly what I've walked you through. We have extended this to be able to pop out of each of those separation lanes, something that we call a gel palette. And this is inspired by some of Professor Nancy Albritton's work now at UW. But to pop out a gel palette from each of these separation lanes that houses, in this case, either nuclei or a single nucleus from the cells that we performed the targeted proteomics on. We do this because we can then pop those nuclei into RT-qPCR to look at the nucleic acids. And we're also working on we're enhancing this workflow to flow into sequencing as well. So that gives us something that is right now a holy grail of single cell biology, which is the so-called multi-mode assays, being able to measure in the same cell the different molecular species that are diverse molecular species. So not just proteins, but in this case, proteins and mRNA. And we have a manuscript under review right now that also includes protein and DNA. So design is important in terms of being able to bring these different functions together. I'm going to skip this. It is published now, some work that we've done on pre-implantation mouse embryos to be able to compare the different blastomeres that form blastocysts, so this pre-implantation mouse embryo, something you could not do if you didn't have microsystems designed to do that. So I'll skip over that because it is published, but I'm happy to answer any questions. In the last couple of minutes, the last topic I wanted to share with you is this idea of being able to not just assay individual cells. So everything you've seen so far are dissociated single cells, suspensions of cells, or even individual nuclei you've seen as well, but asking, can we marry these types of approaches with measurement of cellular context? So put another way, can we do these types of assays, but keeping tissue and adherent cell cultures intact, not necessarily breaking up those cells beforehand? This also was just very recently published, so I just want to share with you the concept and then some of our results, and I'm happy to, of course, discuss, and then also if you'd like to learn more, I'm happy to communicate by email and connect you with the first authors as well. So what do? We have an open device indicated schematically at the bottom. We have those 3 axes, X, Y, and Z. In our case, we perform imaging on adherent cell cultures in that device that are layered on that device. And then we lyse and subject those cells to electrophoresis now in the Z dimension. So we now have a voxel of information, not just a plane of information. And so the goal here is to be able to resolve into the depth of a gel the different proteoform targets of interest. So to use the planar device to be able to measure targets where we do have selective probes, and then to use the Z dimension into this voxel to be able to resolve those different proteoforms and get a readout of the distribution of those in this 3rd dimension. So the device was designed to really overcome some shortcomings with throughput with single cell tools that exist that are typically used for this type of approach. And I'm just going to skip to some of the most important kind of engineering related results, which are number one here, what does our device look like? Basically, it looks like a really thick hydrogel because we now need that Z dimension to be able to perform the separation stage of the assay. And you can see here, Dr. Samantha Grist is holding one of her devices. You can see the stippled microwells all over the top of the device. So we started with isolated cells and then have moved to adherent cell culture. This is what some of those projection separations, so now we're looking in Z, not in XY for the immunoprobing. And you can see this is a confocal micrograph that I'm just rotating to show you the different targets here. In this case, from a purified protein solution, we've done careful quantitation to look at the mechanism of separation to ensure that it aligns with what we expect from theory. We've done some design work, again, to overcome this thermodynamic partitioning that excludes our immunoprobes from the gels. And on the left, you see a nice confocal that shows the concentration of immunoprobe with fluorophore in solution on the top. And then if you look at the bottom, that's our gel. And you can see that because of the size exclusion, the local concentration in the gel is much reduced over that of the immunoprobe in solution, which has negative implications for the immunoassay performance. So we've worked hard to overcome that. And here with just some light sheet microscopy results, have been able to complete and start to image larger regions of interest of these single cell immunoprobes into this, what we're calling projection electrophoresis device. You can see that data with quantitation on migration, and then also looking at endogenous protein from these cultured cell lines that we've started with. We've also developed some algorithms for being able to back out higher resolution information from the separations to kind of recreate the 2D image. We are inspired by the diffraction point spread function, and here in our case we use a diffusional point spread function because we know the identity and the molecular mass of each of our targets because it's a targeted proteomic method, so we can kind of rewind in time back to the imaging plane. I'll leave that there. And this is what those types of separations look like. So we have a throughput of hundreds of cells, and in the Z dimension, we're able to back out information with quantitation about the targets of interest. The last example here from light sheet microscopy, being able to map kind of what the proteoform profile looks like over an XY plane using, of course, that important Z dimension. I think what I'll do is just close with a cryosectioned cell pellet. As I mentioned, we've also looked at adherent cell culture, and this is where we're going as we move towards this sort of projection electrophoresis on intact tissue slices. So with that, I want to take a moment to thank the researchers who conducted the work, our funding support, obviously, which also includes federal and international fellowships, which are an important and unsung hero of graduate education, and then, of course, collaborators, including the patients who were very generous to donate specimens to the research. We do have open postdoc positions, and if you are interested in discussing that, please send me an email at your very earliest convenience, as those are being filled now. And that is it. Thank you so much for your time.
Thank you very much, Amy, and we really appreciate you taking the time to speak to us. This is really
Sure.
elegant work. We do have a few minutes for questions if people want to post any questions in the Q&A box. And while we're waiting for those, I actually have quite a few questions, but I'll hopefully won't ask all of them.
Thank you.
Sure. My first question is actually very kind of a very practical one. How do you pattern the microwells?
Oh, how do we? Fantastic. Yes, I didn't go into that. So the microwells, we typically use an SU8 system. So we basically make, using photolithography, pillars, arrays of pillars, and then just cast the hydrogel polymer around those pillars. So the precursor solution of the hydrogels that we're using, the polyacrylamide gels, are viscosity similar to water before a cross-linking process occurs, and we do that cross-linking around those pillars, and we're able to then just peel off the polyacrylamide from those pillar arrays, leaving us with the negative, which is the microwell system. Does that answer?
Yeah, I think that
Perfect. Great
sense.
question.
And then my second question is, if I understood correctly, you are cross-linking the proteins to the acrylamide between the electrophoresis step and the immunoassay step.
Yes.
Is there any concern that cross-linking affects the protein's ability to bind to the antibodies?
Yes, absolutely. That's a great observation. So it is a covalent immobilization of the target to the hydrogel. It's a 3D hydrogel. As you know, even though in the first examples it's a thin layer, but that covalent immobilization after the separation step and before the immunoassay step most certainly constrains the physical conformation of the protein of interest. Now, we typically use when we perform a sizing assay for that separation step, those proteins have been reduced and denatured. They're fully decorated with SDS detergent. And so they're linearized essentially before the assay. We then anchor them to the gel and use immunoprobes, which are typically developed for linear epitopes on the protein, which means that many of the existing immunoreagents for Western blots, which is a huge market, so there's a lot of these immunoreagents, are quite effective at detecting our protein target of interest. However, anytime you immobilize a complex 3D structure molecule like a protein, you are going to get some artifacts in terms of its conformation. And depending on what your probe is, that might affect your binding. This is true of ELISA plate-based immunoassays. It's true when you fix a cell for flow cytometry and immunofluorescence. And it is also true when we covalently immobilize the cell lysate as well. So an excellent question.
We do have a question from Todd Solchek, who says, good talk. Regarding enhancing antibody diffusion, what active approaches are you considering? Enhanced motion via pressure-driven flow or electrophoretic approaches?
Yeah, great question, Professor Solchek. Thank you so much for coming today. I appreciate your time and your comment as well. So we have been using a lot of electrophoretic transfer approaches. This is an approach that is commonly used with the macroscale systems as well. There's a lot of differences between our system and the hydrogels we're using and the nitrocellulose blotting membranes that are used in the conventional systems. I won't go into that today, but we have found that electrophoretic, that active transfer really does help mitigate this partitioning effect that is just an inherent property of a hydrogel in the systems that we're looking at. We also use dehydration. So again, kind of looking at these passive approaches, so re-wetting of a dehydrated hydrogel with our reagents of interest. And that's been quite effective and some surprising results also that we published there as well. So those are just two of the examples that we've been using. So absolutely. Pressure-driven flow doesn't really work that well with the hydrogels because they have such a high fluidic resistance through that porous material. Those pore membranes are quite small, and so getting an actual flow through them is difficult. But a wicking flow, as I think I just mentioned with the rehydration, does work. Thank you.
Great. Any last question from any of the attendees before we wrap up?
Amy, this is Oliver.
Oh, go ahead, Oliver.
So from the very beginning, how do you load the cells into the wells?
Yeah.
I mean,
Yeah, it's a great question. I made a tiny shout out to that, but I could talk about that for a long time. I won't because we're at the end here. But the results I showed you today are sedimentation, so just gravity-based settling. We do use active approaches, including centrifugation. So some of our devices are fabricated with almost like I need to learn the name of that carnival ride. It's not a tilt-a-whirl, but the microwells are on the wall, and we can use centrifugation to then populate the microwells with single cells. And actually, a lot of the work that we do are in these sparse cell samples. So, for example, the pre-implantation embryos, the circulating tumor cells, because those are such small sample sizes, small number of cells to start with, we can use precision approaches like microtransfer pipetting as well to seat.
Thank you.
Great.
Yeah, sure. If you have ideas on better ways, please let me know. We would love to expand the capabilities for users.
Well, I want to thank you one more time again for a really excellent talk, a very novel approach to this type of analysis. And thank everybody for attending. I remind you, we'll meet again 4 weeks from today on March 23rd with Gary Fetter. And again, I apologize for the issues with the link, the wrong link. So, and that, I think we're going to wrap it up. And thank you again for coming today. We'll see you
It's
next
an
time.
honor. It's an honor. Thank you all. Bye-bye.
Bye bye.